Nature of antigens and antibodies. An antigen is a molecule that triggers the generation of antibodies. The name itself, antibody generator, comes from that definition. The best molecules that are antigenic are proteins and complex polysaccharides. Lipids and nucleic acids do not have good antigenic activity. The area of an antigen where an antibody actually binds is called the antigenic determinant or epitope. One antigen may be large enough to have several antigenic determinants or epitopes. For example, a bacteria could have proteins attached to their surface. Each of these proteins could have several antigenic determinants or epitopes. Here, for example, is an antigenic determinant which is bound by this antibody B. Within that same antigen, there is another antigenic determinant, in this case bound by antibody A. So different types of antibodies could be bound to a bacterial cell carrying antigenic proteins or proteins on their surface which are antigenic, because each protein has different antigenic determinants. The B cell, which generated the production of antibody B, would be different from the B cell that generated the production of antibody A. Antibodies are proteins made by two chains of amino acids. One continuous chain is longer than the other and is called the heavy chain. Each antibody molecule has two heavy chains. Here is one, and adjacent to it is the second heavy chain. There are also two shorter amino acid chains called the light chains. Here's one, here's the other. These are the light chains of the antibody. Both heavy chains are connected, as are a heavy chain and a light chain. Each heavy and light chain have a variable region at the end of the chains. This is the area where the antigen is going to bind. So the variable regions of an antibody are the areas where the antigen bind. The constant regions of the antibody will be the area that determines the class of the antibody. The amino acid sequence in the constant region of the heavy chain are the same for all antibodies of the same class, as are the amino acid sequence on the constant region of the light chain for antibodies of the same class. What determines the specificity of the antibody or the type of antigen the antibody will bind will be the variable region. There are 5 classes of antibodies, which means there are 5 different types of constant regions: IgGs, IgMs, IgEs, IgAs, and IgDs. IgMs are the first antibodies to respond to an infection. These antibody molecules, arrange themselves in a pentamer fashion. This is the only antibody that can be formed by the fetus. Again, is the antibody that is first made during an immune response. IgG is the most abundant antibody in circulation. This structure is that of a monomer. It is the only antibody that crosses the placenta and will be given to the fetus. It is the most specific. It's the best of all antibodies. It is the antibody made by memory cells. IgA is the antibody found in secretions, in tears, saliva, mucus, breast milk. It can be found as a dimer in secretions, as a monomer in serum. IgD is very rare in circulation. It is typically attached to B cells and is involved in the maturation of the antibody response. IgE, again very rare in circulation. This is the antibody responsible for allergic reactions. Here are all 5 classes of antibodies with their molecular structure. IgGs, IgEs, and IgDs are monomers. IgAs are dimers. IgNs are pentamers. You now should be able to explain the characteristics of antigens and determine which bacterial structures could be antigenic. Remember that antigens are proteins or complex polysaccharides. So cell walls of gram-negatives, cell walls of gram-positives, flagella, capsules. If these structures have proteins or complex polysaccharides, they will be antigenic. The nucleus of a bacterial cell. Again, if there are proteins or complex polysaccharides, that area will be antigenic. The nucleus will be the area where nucleic acids are found. You should be able to describe the molecular characteristics of antibodies, which part of the antibody molecules determines its class, the constant region, the variable region. List the 5 classes of antibodies and give the molecular structure of each. Are they pentamers, dimers, monomers? You should be able to match an antibody with its characteristics, which is the most specific, which can cross the placenta, which is found in body secretions, which is the antibody made during the first primary immune response, which antibody is secreted in greater amounts during the secondary immune response.